Good afternoon, everybody. Welcome back after lunch. My name's Kinjal Majumdar. I'm an assistant professor of molecular virology and oncology at the McArdle Laboratory for Cancer Research, and I will be chairing the first half of the afternoon session. So it's my honor to introduce our first speaker this afternoon, Dr. Lou Mansky. Dr. Mansky has had a long-standing interest in the replication, evolution, and virus-host cell interaction of human retroviruses, including HTLV-1 and HIV, both 1 and 2. His laboratory has been involved in HIV mutagenesis, antiviral strategies, comparative analysis of HTLV and HIV particle assembly, and host cell interactions. Dr. Mansky is the founding director of the University of Minnesota's Institute for Molecular Virology, as well as the founding director of the NIH T32-supported Minnesota Training Program in Virology. And today he's going to be talking, the title of his talk will be, "You Can Observe a Lot by Watching, New Insights into Human Retrovirus Particle Assembly by Comparative Analysis." Let's welcome Dr. Mansky.
Thanks, Kinjal, and I want to thank the two Pauls and the entire organizing committee for the honor and privilege to be able to speak to everybody today. Before I start and dive into the science, I wanted to talk a little bit about the more personal side of Howard as a mentor, and I'm going to do this in two short little remembrances. So start out on my first day in the Temin lab. First day I came in, went into Howard's office, and he wrote out, kind of how the lab works and everything, and he gives me a list of things. I need to go upstairs to the 10th floor and talk to Karen Schwarz and do this, do that, and I take this, he wrote it out for me, and so then I came back from his office and then went into the student office, which was right next door, and in that era, the people that were sitting in there were at the desk I sat in, to the left was Winnie Patak, and to the right was Wei Su Hu. So I went to Wei Su, and I showed her this list that Howard had given me. And the one thing I was puzzled about was that he had indicated to me that on Friday I would meet with him in what he called research conferences. And I said, "Wei Su, what are research conferences?" And she said, "Lou, I have no idea what that is. We have confessions on Friday." And so let me tell you a little bit. So I don't know this for a fact, but I believe that John Coffin is the one that coined confessions. And I'm assuming that Howard knew that we all called it the confession, but it was never acknowledged. There were a lot of things that weren't acknowledged to Howard. There was a lot of things that weren't communicated, that was just assumed or was inferred. So in my time in the lab, so I was in the lab in the early 90s, 1990 until Howard's passing. So I was there during the era when both Rex Surser and Howard passed away. That was a tough time in McArdle. I'm grateful to not only having been mentored by Howard, but I'm also grateful to the mentorship of the entire McArdle laboratory, because that was a real difficult, challenging time. There were a lot of things to be learned as a young scientist in terms of career development that happened during that time, and I'm grateful for both the era when Howard was healthy and the era where he had to manage a life as a scientist and as a husband and a parent dealing with a life-threatening illness. That this was not a challenge, but he found a way of finding a way of getting back into some routine, and I watched that entire time. So I would normally get in, at least as a student or a postdoc, I was a little unusual that I came in fairly early. I would get in before Howard. Howard was pretty timely about getting in at 9 a.m. And he would either ride his bike or he would walk in from his home. So I would typically get in around 8:30. I just lived not too far from here, a couple blocks away. And Howard's office was unusual, and I still think this is kind of crazy that his office was. So his office was a lab, right? There was a lab bench in his office. And there was a front door where you walked in, but there was also a back door. And that back door was never locked. It was an unlocked door. And he obviously knew this. And I was always kind of puzzled that when he left and at the end of the day, he would close the door to the front of the office and he'd go home. But the back door was open. And when I joined the lab, people just went into his office. And what was the only unusual thing about his office was that there was both a Mac and a PC computer. And in that time frame, in the early 90s, that was really unusual that a faculty member would have both two computers, but they were different operating systems. So he used the PC for word processing, he used the Mac for graphics. And it was understood to me and others that you could come in and use the computers because there weren't that many. It's crazy to have to admit this, but there weren't like a whole lot of computers around. There weren't like laptops or anything. And also you could use office phone to make long distance phone calls. And so he had a little sheet there where you would, like a little spiral notebook, teeny tiny one, that you'd write down, who you called and the date, so that if there was a discrepancy or some issue, like some crazy call to Australia for 2 hours or something, go back. And so anyway, so I would normally meet with him in the morning because I was there, and he, as Mike Emmerman mentioned, that he would walk around and greet everybody in the morning. Sometimes he would go through the back door because he knew my lab bench during my time in the Temin lab was the first lab bench right next to the back door of his office. So a lot of times he would peer through and take a peek to see if I was there, and he would greet me and he said, "Do you want to talk?" I said, "Sure, I'll go ahead and talk." So many confessions for me were in the like first thing in the morning. So come 9:05 or 9:07, I'm done. I'm done for the day. No more confessions. I could, you kind of felt like you could relax for the weekend because you were done talking for Howard a week. I shouldn't say that, but you felt a little bit more comfortable working the rest of the day. So there was one time, this is now, I'll take you back, it's probably mid-December of 1991. So in 1991, they had a Nobel Jubilee, and this was for the 90th anniversary of the Nobel Foundation. And he must have went there around like the 10th of December. That's when they give out the Nobel Prize, which was also Howard's birthday, and Howard was awarded the Nobel Prize, I believe, on his 41st birthday. So that year it was the Nobel Jubilee. He went to that, he came back, and so that was in the afternoon. So I didn't talk with him in the morning, it was in the afternoon. I met with him, it was after lunch, went to his office, and we talked. I actually don't remember exactly what the research update was, but as I'm walking out of his office, so his desk was facing towards the front door, so you come in, sit with him, and then you turn around and walk out. As I'm walking out, he goes, "Oh, wait, I've got something for you." And he got up, went around his desk, he went up, and up towards the front, below a table, he had a small dorm fridge. He reached in the dorm fridge and pulled out a gold-foiled chocolate Nobel Prize. And he gave it to me. He goes, "Here, you've got your own Nobel Prize." And so my response to him, he caught me totally off guard. Like, are you kidding me? So there were times where I would definitely joke around with him, and he was totally okay with that. It wasn't every day, but certain times you could kind of joke around with him, and he was totally okay with that. So my response to him was that, "Well, Dr. Temin, people have always asked me where you keep your Nobel Prize. I can now tell them that you keep it in your refrigerator." So he looked right back at me. He immediately responded. He goes, "Do you want to see the real one?" And I was like, I don't know what? I said, "Sure." So he went back behind his desk, and in one of the file cabinets, he went all the way down to the bottom drawer, opened it up, pulled it all the way out, and he reached way in the back, and he comes out with this box. And he came back up to me. He opened it up. It was the Nobel Prize. So then he explained to me that when you get the Nobel Prize, you get two of them. One of them is gold-plated, and he said, "This is this one." The other one is the solid gold one. He goes, "Of course, that one is kept in a safety deposit box." And it's like, I can't believe you just did that. So then I walked out with him and went into the student office, and I don't remember who all was there. I don't know if there's anybody that's here, any alumni that remember that, but he walked into the student office and just showed people, whoever was there at the time, the Nobel Prize. And then after that was over, he put it back down in the back of the file cabinet, and it was the end of that. I have no idea how many times they did that. It was obviously out of character for him to that was not his style. He's a scientist. It's all about the science. That's what we all learned from him. But the thing is, he was a human person, and you could joke around with him, and he could be playful. But in the end, it was about the science. But he was an honest, decent person that you could interact with on a personal level and connect with him. And a lot of people I've met over the years did not see that part of Howard. They just saw the tough, rigorous scientist. What they didn't realize was that was the other side of him, the human side, that I just found was just so joyful and so pleasant. And I'm so honored and privileged to have trained his lab, because it helped me a lot as a mentor myself. So I wanted to share that with you before I now dive into, kind of nerd retrovirology. So you'll have to go along with me for the final few minutes here of my talk. So what I'm going to do is just give you an overview of a couple of projects. One is related to answering a scientific question. The other one's going to be related to trying to address a technical issue. So let's give you a little bit of background because it's a fairly diverse audience. So our lab studies the human retroviruses. Many people have already heard about the HIVs, and most of you now know, all of you know something about HIV, so I don't have to belabor all the details about HIV, but I get to introduce to you human T-cell leukemia virus, type 1, which is the cancer-causing human retrovirus. And 2 points that I'll just bring up on this slide. If I can use my laser pointer. You can see that it actually, the reason why most people, and I have to tell students this all the time, gonna move my little laser pointer down there. Most people have not heard about HTLV1. The reason why is that it doesn't have a high kill rate. So it's those who are infected with HTLV have been abandoned and orphaned by biomedical research because there's just not enough of them. And there have been times where I've talked to students about it. It's like, well, why do you really care about HTLV? It's like, well, I've never said this, but I'm thinking in my mind, it's like, well, how many people have to die before this is important? What's the number of people that have to die of a viral cancer before that becomes important? I don't know what the answer to that is, but it's generally not, it's been somewhat forgotten because it has a low rate of death caused by the leukemia and lymphoma. The most interesting part of the biology of this virus is that it's really highly dependent on virus spread by cell-to-cell contact. And when you think about viruses, this is really an unusual quality that a virus would have evolved to persist in nature by needing to be in direct contact with target cells in order to be transferred and maintained in an individual and be able to be transferred from one infected individual to another. I don't have a good answer for you as to why that is, other than it's an unusual evolutionary lifestyle that the virus has chosen to be able to persist in nature. So let me give you a little bit more background about HTLV related to this cell-to-cell spread. And there's been many publications in the field related to different types of biology, cell biologies that allow for cell-to-cell contacts. And because of this mode of spread, cell-to-cell spread, it still means that it's a virus, right? What viruses do is that they infect cells, but they have to create progeny virus. They got to assemble and produce a virus, and then they go through these various pathways in order to be spread to a new infected T cell. So it's an unusual lifestyle, but this is how this virus has adapted and evolved to maintain itself for over 100,000 years in the human population. So assembly actually is important. And like Eric Freed, we also are interested in studying the later steps of the retroviral life cycle, and shown here is the later steps in the assembly pathway. And what I'm going to do now is this is a cartoon. I lecture a lot to students, and I always tell the students this, that when you see a cartoon, you should be immediately suspicious, because usually the virologists show the cartoon because they don't fully understand what's going on, so they draw a cartoon instead of just showing you the data. But what I'm going to do now is to first show you some data from a project that will answer a question. I don't know if I can get my laser pointer. So I'm going to talk about this I'm going to actually go backwards, looking at immature virus particles and comparing their morphologies, and then I'll deal with a technical question that relates to the multimerization of this GAG protein. So GAG is the major structural protein of a retrovirus. And that'll be the main viral protein I'm going to tell you about. It's got 3 major domains: matrix capsid, nucleocapsid. So I like showing the slide, and I'm going to actually advance it one more so you can see some additional data here. So this was a study that was done by a former PhD student in the lab, Jessica Martin. And essentially what she did was to basically do a comparative experiment to line up as many different retroviruses as possible and produce immature virus particles so we could compare the immature particle morphologies by cryo-electron microscopy. And she was able to get the majority of the viruses purified and concentrated enough to be able to do this kind of analysis. The exceptions were beta retrovirus and an epsilon retrovirus, so those are missing here. So blown up here, you can see that there are HIV-1, HIV-2, and HTLV. So if you look at, this is not always that easy to see, so that's why I'm showing these cartoons here. But let me first explain this briefly. So what you're looking at, this is an immature virus particle, so the density on the outside is the viral membrane. Right below that, this density is the gag lattice, the immature gag lattice, and then this density inside could be nucleic acid, other things that have density. So if you look at the HTLV1 particles, you can see there's gaps in the lattice, and it's not really very complete. That's kind of depicted here by other types of studies using cryo-electron tomography and subtomogram averaging to give you an idea of, so these green hexamers are 6 gag molecules that arrange in a hexameric arrangement, and then the hexamers interact with each other to form this gag lattice. So you can see that it's almost like islands of gag lattice that wind up building a HTLV1 particle. HIV is a little bit better, a little bit more coverage, and then HIV-2, we like to describe it as being quite beautiful because it almost forms a complete gag lattice without virtually any gaps in the lattice. So you see they're all different from one another. The one that was the most interesting and intriguing to us, I'm going to show you data from HTLV1 and HIV-2, but HTLV1 has always been the most intriguing one because it's so different than all these other retroviruses if you looked real carefully. So the goal was to go from those 2-dimensional, low-resolution cryo-EM images to high-resolution, because one of the things I always appreciated from Howard and why I was so honored to train with him was his interest in quantitative virology, getting into the details and really figuring out what's going on. So that's the one thing that you can do now with the high-resolution cryo-EM. This is pretty exciting, but I don't know if it's exciting enough to have all the stuff going on in the background. So I don't want to get into all the details. This is if you can get high-resolution structural images from these immature HTLV1 particles. The upshot of these studies were that the capsid N-terminal domain possesses the main interaction residues that help build this lattice. So the capsid domain for most retroviruses is known as being the main domain within the gag polyprotein to form these gag-gag interactions that lead to the hexameric arrangements that then lead to this lattice sheet being formed. So with HIV-1, those main residues are actually in the C-terminal domain. For HTLV-1, it's actually in the N-terminal domain. This is another interesting evolutionary question as to why did this virus, that's a highly dependent on virus spread through cell-to-cell contact, basically reverse the orientation of where the critical amino acid residues are to allow these arrangements to be formed to actually build an immature virus particle and move it to the opposite side of the protein. So this remains an open question, but it took many, many years to get into the molecular details of this to actually prove that this is true. So of course we have a lot of site-directed mutagenesis data to demonstrate the same thing, but structurally, this is how this works for this virus. It's actually fundamentally quite different. The other things that you can do when you're doing high-resolution cryo-EM is that the subtomogram averaging allows you to, and this is a study that was done by Bill Arndt. Bill is here today, he's sitting in the back. He has a poster, as well as several other students from our lab at the University of Minnesota who are out there as poster presenters. I encourage everybody during the break to stop talking for at least a couple minutes and please go and visit these posters if it intrigues you at all. So this is something that Bill had done to be able to look at much more detail of these hexameric arrangements, including figuring out the details of this flattened region, which is a highly unusual feature in retroviral immature particle morphologies, is that you see this flattened region. And you can also determine how far away from the plasma, the inner leaflet of the plasma membrane that hexameric arrangement is, as indicated here. He's able to get high-resolution structural information down to 3.4 angstroms, which at least in situ in a virus particle, this is the highest resolution to date for HTLV1. And he's showing this flex refinement here down at the bottom. And this is what we think is the explanation for why a flattened lattice can actually form versus the curved lattice that would carefully follow the viral membrane. This is looking at HIV-2 now, where you can see this very complete. This is the high resolution structure. This we only get down to 5.5 angstroms. This is showing the near completeness of the immature lattice. And this is a movie that was created by my collaborator, Wei Zhang, who's actually here as well from Minneapolis, showing what it looks like in 3 dimensions. This is also really unusual too. It looks very, very different than what you see with HIV-1. Also curious as to why this closely related HIV looks fairly different in terms of the recruitment of GAG. It asks really fundamental questions as to what's driving these differences with these different retroviruses that are clearly all close cousins to one another. Just a little bit more detail before I switch to the last topic. That is the key residue. So we were trying to figure out the differences between HIV-1 and HIV-2, and we think that part of the answer is in this interaction between helix 10 and helix 12 of the capsid domain. We made mutations here. At least we were able to show that this can affect maturation, where if you make a mutation, you eliminate core formation infectivity. We also think this is the region, too, that helps to explain why you see increase in GAG copy number. So I'm going to switch and just talk about the technical issue that we need to solve. So this is just showing a movie of looking at live cell imaging of producing virus particles. So if you tag the GAG protein with a fluorophore, you can produce fluorescently labeled particles, but you can also look at the trafficking into GAG. And so we have methods where we can do this at single molecule resolution. We've done studies where we've tried to figure out where does the GAG come from to get to a virus, asking a really fundamental question. When retroviruses assemble, how did they figure out where on the plasma membrane we're going to set up shop and build a virus particle? That's the basic question we're trying to ask. So we've seen with HIV-1 and HTLV-1 that they fundamentally recruit GAG by 2 different methods, either directly from the cytoplasm or GAG goes to the membrane, it moves along the membrane, finds the virus assembly site. When you're doing that, you have a problem, though, is that because what we're really interested in is trying to find out where the productive particle assembly sites are. It turns out most assembly sites are not productive. They don't produce virus particles. It's actually a low-frequency event that they actually form particles and produce them. So if you're doing these kind of live cell imaging experiments and you're looking at the bottom membrane that's up against the cover slip, you can't look at particle release because you're on the bottom cover slip. So you need to look at the top membrane. Most imaging methods, that's a problem. So this is one method that you can use to address and look at events at the top membrane. It's called lattice light sheet. And you can see here, if you look at the bottom membrane, real quickly, this is the second last slide. If you look at the puncta that you're seeing are GAG puncta, and they're actually assembly sites. If you look in this, there's a lot of stuff going on, so don't get too confused by that. Just look in the red boxes. On the bottom membrane, they're not moving, they're stuck. Whereas on the top membrane, you can actually watch it in the movie, you can see that they're actually moving along the membrane. This is a dynamic that you don't ever really see on the bottom membrane. It's telling you there's a different type of cell biology going on at the top membrane that you don't see on the bottom membrane. The other thing that was noticed was that the assembly rate, the rate at which GAG is being recruited to the assembly site, is actually much more rapid down at the top membrane than at the bottom membrane. So the assembly rate is very different. The fundamental nature of how the virus is assembling is actually different depending on what membrane you're looking at. So in conclusion, what I just showed you is that there's a distinct nature of GAG-GAG interactions that help to explain differences in the morphology of HTLV1 particles, and that might likely be true with the mature ones. I showed you some of the data with HIV-2 and showed you how the live cell imaging techniques that we're using that can help to address the question of figuring out where the productive virus particle assembly sites are. So I want to acknowledge people in our lab. There's actually one person that's here that I apologize. Your name is actually not on the slide. Sorry about that. Our collaborators, I really want to, I'm an interactive collaborative scientist. Wei Zhang it helps us do the cryo-electron microscopy and tomography. We simply could not do this science without her. Joachim Mueller is a photonics expert that helps us with doing the super resolution microscopy and the various techniques. I also want to point out too that I know many of the current UW virology faculty, as well as many of those that are also in McArdle, because I've hosted with Paul Lambert the Whiskey Soda Symposium, which is over here. So this is coming up just in a couple of weeks now. So Paul and I have partnered to do this for 13 years now, and that's also been a joy and a pleasure, but also an honor and a privilege to be able to connect with people here in Madison in the virology community to work together to try to improve the career development of young virologists and training. So with that, I will thank you, and I'll be happy to, if there's time to answer any questions.
Okay, we have time for only one quick question.
So your HTLV particles are really intriguing in the sense that there's a really large gap between the membrane and the GAG lattice. Have you been able to visualize the matrix lattice in those particles to see if they're pulled away from the membrane or if matrix is more flexible for HTLV and is being, made less globular, more linear? Have you looked into that?
We have looked into that. I actually have the answer for you on a slide, but I ran out of time. But I can talk to you later about that. But we don't have the full data, but I can at least provide it. The explanation is actually discussed in Bill's manuscript that we're hoping to be accepted any day now. But so at least we have an idea of what would help to explain that difference. But we have not, and we're obviously interested in looking at the matrix protein in terms of its structure. But I have an answer for you.
Yeah, great, thanks.
Let's thank Dr. Mansky.